Thanks for the introduction, and thanks for staying after the keynote, and also thanks for the program chairs for giving us the chance to present our approach to the ENCODE Dream Challenge. And this challenge has been about the computational prediction of transcription factor binding as we can measure it by ChIP-seq experiments in new, previously unseen human cell types or tissues using a minimal set of experimental assays and doing that on a genomic scale. And more specifically, the setup of the challenge has been that we were provided DNA-seq data representing chromatin accessibility. We were provided RNA-seq data about gene expression, and for training cell types, we were also given ChIP-seq data specific for several transcription factors, and derived from that labels about a region on the genome being bound or being not bound by that transcription factor or being ambiguous. And the task of the challenge then was to predict labels on previously held out chromosomes in previously unseen cell types on those chromosomes. And what we were allowed to use additionally were motifs from any source, the genome itself, of course, and gene annotations according to GenCode 19, and also computationally derived DNA shape parameters. And this is an overview of all the cell types and transcription factors we were using. We had 109 training combinations of cell types and transcription factors. We had a few that we could query in a leaderboard round to get an impression of our performance of the current model. And we had 13 data sets for a final submission round that were then used for scoring the challenge submissions. And as you may appreciate, most of those transcription factors for the final submission round have been measured in a liver cell type that never appeared in the training data and also appeared only once in the leaderboard data sets. So what we first thought about in approaching the challenge was what features we could use. And what might be quite obvious is that sequence motifs of the different transcription factors might be informative about binding regions that could be measured by ChIP-seq. And so we used motifs from different databases, the Hokomoko database, our own in-house database, dbCore-DB, and also from Epigram, which represents something like histone modifications, or at least motifs that are indicative of histone modifications. And in addition, we used our own motif discovery approach to learn regulatory motifs from the ChIP-seq data that were provided in the challenge, and also from DNase data. And if you would like to try this out, that approach is available as a command line application and also in our Galaxy instance locally in Halle. In addition, we used several features derived from the DNase data provided, short-range and long-range statistics, and also statistics about the fluctuations or the monotonicity of DNase profiles. And finally, from the RNA-seq data, we used the TPM value of the closest gene for the current region for which we should make predictions. And we also derived several sequence-based features, information theory-based features, and something like GC content or number of CPG dinucleotides, things like that, and also annotation-based features. And in the end, we should make predictions for a complete genome, and we could compute all those features in a one base per resolution. But the memory requirements and also runtime requirements for making predictions in the end on that basis would be just too large. So we thought about ways of reducing memory requirements and as the original challenge requested us to make predictions in 200 base pair windows, shifted 50 base pairs by 50 base pairs over the genome, we decided to bin the complete genome into such 50 base pair regions and just compute aggregate statistics in those 50 base pair bins and use those for learning a classifier afterwards. And so a training region for us consists of those aggregate statistics for several consecutive bins on the genome. The model we use for representing those features is quite simple. It's just an independent product of Gaussian distributions for all the continuous features and multinomial distributions for the discrete features. And as a learning principle, we use a weighted variant of the maximum conditional likelihood principle, which is somewhat related to logistic regression, but imposing those distribution assumptions. And again, we had the problem that the number of negatives by far exceeds the number of positives, so the number of regions that are indeed bound. And we didn't want to use all those negatives as a negative training sample. And we thought about different ways of subsampling the genome and tried different strategies and came up with a sample along the genome that should be representative, which we accomplished by weighting that sample, but also should contain complicated cases like regions that are open chromatin. And we did an initial training using that sampled negative set, and it didn't work too well, actually. And we were not really satisfied with the performance. And so we came up with an idea that we call iterative training, where we first used that initial negative regions and the positive regions that are known to be bound by the factor, and we learn a classifier from that. And then we used that classifier to make predictions on our training cell type. And for the training cell type, we know which regions are labeled to be bound, which regions are labeled to be not bound, and which are labeled to be ambiguous. And now we search for new negative examples that are hard to classify for a current classifier, which means that the probability of being bound in those regions is very large. And we select such regions and augment our negative training sample by such regions that are not classified well by our current classifier. And then we retrain. And we do this for 2 rounds and now have, for one training cell type, one training sample, 3 independent classifiers that have been learned on those iteratively augmented data sets. So how do we make predictions in the end? We have the genome binned into those 50 base pair bins, and the training regions have been consecutive bins, and now the regions for which we make predictions are consecutive bins as well, centered as at the bin for which we want to make the prediction if it is bound or not bound. And this is represented by the classifier for cell type J. The kth classifier of those 3 we learned is a probability for that specific bin and that classifier. And we can compute those probabilities for all the bins along the genome. And if we now want to make a prediction for one specific bin, we compute the probability that at least one of the bordering regions for which we made predictions overlaps at least half of that region. This has also been the criterion for defining the labels initially from the ChIP-seq data, so this is consistent with how the labels have been derived. And we now have different classifiers from the iterative training procedure, and we have different classifiers for the different training cell types. And our final prediction score is just the average over all those derived probabilities. So how does this work? We first assess performance by the area under the ROC curve and got a really nice picture. So for all cell types and all transcription factors, we get areas under the ROC curve above 0.95, and this should be a satisfying performance. Perfect classifier gets an area under the curve of 1, so we are scoring quite well. Do we really? Well, the problem is we have highly imbalanced data. So we have a ratio of positives to negatives of 1 to 100 in the best case, and far exceeding that number, so the ratio of positives to negatives for most of the factors. So the area under the ROC curve might be a bit misleading. And if we now look for the area under the precision recall curve instead, we see that performance is not so great as we thought initially. So there are exceptions like CTCF, where we still score quite well, but for the majority of transcription factors, we are way below 1, and so there is still much room for improvement. And if you would like to also assess your imbalanced classification problems by the area under the precision recall curve or the curves themselves, you could try our package, which is available from CRAN. So after that, how do we score on the complete challenge? We submitted our predictions, and this is the result, which is the mean normalized rank, aggregating several performance measures. And in the end, we came up at rank 1 together with another team, Yuanfang Guan. So although we were not really satisfied with the performance on the level of AUC-PR, at least this seems to be as good as it can get at the moment. So after the challenge was over, the test data sets were published, and we were able to look a bit deeper into which of the features we used are really informative for our predictions. During the challenge, we just kept adding features and didn't really have the chance to test them individually. And so what we did was we tested our model using all the features and also tested the model excluding a specific group of features. In this case, we excluded all features that are related to the DNA-seq data. And we then compute the difference of those performance values, and we represent this in a compact form as such a violin plot. And we did that for different groups of features, and what we see is that DNase data are the most important feature for making genome-wide predictions of transcription factor binding. So chromatin accessibility tells us a lot about if a specific motif on the genome is bound by a transcription factor or not. The motifs also help in almost all of the cases. There are some notable exceptions where adding motifs to the model actually leads to a decrease of performance. And we also tested if the motifs we learned by our motif discovery approach from the training data help, and indeed they do. And we also tested if our models capturing intramotif dependencies have a contribution to the final performance. And yes, they have, although the contribution is small. So adding intramotif dependencies adds a little over simple PWM models, but the contribution is not too large. And we also tested the RNA-seq-based features, annotation-based features, and sequence-based features. And at least in our model and how we do it, they are basically useless. So then the next idea would be if those features are all largely useless, why can't we come up with a model that just uses DNase features and motifs and ignores all the others? And we tested that model combining DNase data and motifs as well and compared it to the model we used for our final predictions, and we found that they perform quite comparable. So this also means that we now only need one experimental assay, which is DNA-seq, and we can skip, at least in our model, the RNA-seq data and can make quite accurate predictions using just DNA-seq data and motifs learned, and a model learned from those training cell types. So with that, I would like to acknowledge a few people. First of all, my other fellow team members, Jens Keilwagen and Stefan Posch. And I would also like to thank the challenge organizers for setting up such a nice challenge and fixing performance measures in advance and really doing this very rigorously. And yeah, it was just a nice experience to participate in that specific challenge. And I would also like to thank members of the other teams which shared their thoughts and their ideas about features one could use in the conference round very openly. And I think the improvement we observed from the initial conference round to the final submission round also heavily depended on adapting ideas of features we could use that were inspired by work of those groups. So with that, I would like to tell you what I would like you to remember from my talk. And first of all, the prediction of in vivo transcription factor binding, as we did it in the challenge, seemed to mostly depend on chromatin accessibility and binding motifs. And it might be that other features that were not allowed to be used in the challenge could still add something to the performance we observed. The prediction so far works well for some of the transcription factors, but there's still room for improvement, and we are not yet at a level where we could replace ChIP-seq experiments by computational predictions. What I would also like to emphasize is if you have imbalanced classification problems and you want to assess performance, use the area under the precision recall curve, at least in addition to AUC-ROC when assessing classification performance. And what I also found notable in the challenge was that, at least among the participants, deep learning approaches didn't outperform old school machine learning based approaches like logistic regression. So the both top teams used something related to logistic regression, and there have been some deep learning approaches that didn't score too well in that case. And finally, I would like to encourage you to participate in challenges, because they are a great opportunity to get an unbiased and fair comparison to other methods, and also greatly foster collaborative science. So if you have the chance, do it. Thanks for your attention.
Thanks a lot. While, and I'm assuming that all other methods must use these features plus everything else. So why? What's your intuition why you could do much better than others?
I think one thing is which features you derive from the DNase data, and what we initially used was just statistics of the coverage profiles and peaks. And in the second round, we also used something about the continuity of the profile, which has been inspired by a feature derived by autosome.ru. And this helped a lot, I think. And also the idea to derive motifs from the DNase data, not only from the ChIP-seq data. I think that also helped a lot. And finally, the iterative training also helped, I think.
Yeah, thanks for a nice talk. I was surprised that that your de novo motif search did not perform better than the motif databases. At least it looked like that from your delta analysis. Do you have a remark on that, why that is so?
Yeah, I could comment on that. So One thing is that we used our in-house database, which contained several motifs learned by our approach from ENCODE data, but for other cell types. And this also includes, to some extent, more complex models. And so what you could learn from the additional training cell types are maybe cell type specific motifs or variations of motifs, but the motifs in general are already represented, learned by the same approach in our database. And so this might be the reason why we don't see a huge improvement by de novo motif discovery in this case.
Very interesting talk. I have a quick question. If you did you use all the motifs available in all three databases, or did you pick and choose? For example, for CTCF, did you pick a certain number of motifs?
We picked motifs that have been annotated for those transcription factors, of course, and we also selected some motifs that are known to, of transcription factors that are known to interact with those we are interested in. And we also did some analysis regarding this on the training data sets. So something like overlaps of binding regions and things like that to select additional motifs of other factors.
Hi. What are the continuity-based features? Are they using information from the flanking bins or something?
I didn't.
What are the continuity-based features that you talked about? So from the DNA-seq, not just the motifs you also used. Yeah.
Yes, it's something like for how many steps does the profile just go upwards or downwards, and how long such continuous monotonous stretches are, for instance. So it's something about, do we have large fluctuations and it's going up and down all the time, or do we have something that is shaped like a peak, basically?
Hey, nice talk. So my question is regarding the DNA's data and how it helps a lot in the final performances, especially the AOPR. So if that contributes most of the final scores, what is the challenge right now with the bound regions that don't have DNA's accessibility data? Those simply cannot be predicted with any of the other features? Or what are the directions, for example, in getting accessibility data with other data sets such as ATAC-seq or trying to somehow learn maybe from a different approach, not necessarily DNA's data?
Yeah, I'm not fully sure about that. So we had some examples of transcription factors that also occur in regions that are not accessible or that do not correlate well with the DNA's data. And in those cases, we saw that motifs help a little more. And yeah, DNA's data helps less. But in the end, this is still something where you could learn about shapes of the DNAs or something like that. But we didn't test our approach for other methods like ATAC-seq or something like that so far.
Thank you.
Hi. Just out of curiosity, you were saying that there were some motifs that were like diminishing your prediction ability. Do you remember which were they or any biology out of them?
Yes, so I think the most prominent example has been JUN-D, where we observed that. And we speculated a bit, but we didn't fully untangle the reasons for that so far. So one speculation is that the motif is just very short, very conserved, and so it occurs basically everywhere in the genome, and this is something that doesn't help in the end. Another option could be that the combination of a transcription factor with some cofactors is highly cell type specific. And as we learn about the motifs from training cell types and then make predictions on another cell type, it might be that what we learn about motif combinations just doesn't hold for that prediction cell type. And so what we learn from the training data sets in that case would be just misleading. Some kind of overfitting to the training cell types.
Okay,
just to update everyone, so the next speaker, apparently there's a visa issue, so that's why the person's not here. We're going to take a break until 11:30, and then we'll reconvene at 11:30 for the next speaker.